Standard 4 for the cells unit is about the parts and the function of the endomembrane system. So the last standard, there was only two parts, the nucleus and the ribosome. This one, there are a few more, but they're less stuff to understand. So the nucleus and ribosomes are pretty much like the most important parts of the cell, but there are only two. This one, there are lots, but they all have like a very, very small function. So what does the system actually do? What do you mean by the endomembrane system? Well, what it does is it regulates the traffic of protein, and it also deals with the metabolism of a cell. So here's the nucleus we were talking about, here's the ER we were talking about, and now we're going to talk about this system going from the nucleus to the outside of the cell. So what does that system look like? That's why they call it endomembrane, from basically the nucleus to the outside. Well, the very first thing we're going to talk about is ER. We just briefly talked about in the last video. There are two types of ER. There's called rough ER and smooth ER. ER is basically a network of membranes and sacs. Another way to think about it is like a freeway between the nucleus and the outside of the cell. So the ER is like a freeway. The rough ER has ribosomes on the surface. We talked about this in the last video. So they're like little dots literally on top of the ER. They package proteins for secretion. They send these transport vesicles, or they call them like transport... vesicles is like think about it as like a transport, like a delivery truck, to the Golgi apparatus, and they make replacement membrane. So membrane, remember, is like the shell, so rough ER makes the replacement. Smooth ER has no dots, has no ribosomes on the surface. What it does is that it creates lipids, metabolizes carbs, it detoxes the cell, like if there's drugs or poisons or things that the cell doesn't want, it detoxes, and it stores calcium. So you need to know the difference between rough ER, rough ER meaning like they do the proteins and they make replacement membrane, versus smooth ER, which has no ribosomes and they deal with like lipids, carbs, drugs, poisons, stuff like that. Here's another picture of it. Remember, this is my nucleus, the purple. Here's my ER in blue. The smooth ER, as you see up here, have no dots on it, and the rough ER do have dots on it. Here on the TEM, this little canal, these rivers with dots on it, that's rough ER, versus this stuff right here, which is the smooth ER, which has nothing on it. The second part of the system is called the Golgi apparatus, or in some other books they call it the Golgi body. So if I look back at this picture, I have my nucleus, which then goes to the ER, which we talked about, and then we're going to talk about this little big old green thing, which is called the Golgi body, or the Golgi apparatus. The whole reason for Golgi apparatuses is think of it as the UPS of the cell. All it does is create and package molecules. They use it for transport in vesicles. Remember, think of it as like trucks. And they produce lysosomes. So the Golgi apparatus, this green thing right here, its whole job is to package stuff. It's a series of flattened membranes and sacs called cisternae. So cisternae is the name of the membranes in these sacs. The cis face is the receiving side of the Golgi apparatus. Again, think of this as a UPS. So it receives the stuff from the ER. You guys see these little dots right here? So these are the vesicles. Like a vesicle, think of it as a bubble. It comes in and it packages. It's like, here's something I want to ship. Can you package it for me? So the cis face, this side, receives vesicles. The trans face is the shipping side. So after it's done packaging what you want to get rid of or send out, the trans face then sends it out. Here's a picture of the Golgi apparatus through a microscope, electron microscope. When we talk about how they produce lysosomes, what is a lysosome? Well, the easy way to remind my former students of what a lysosome does is think of lyso as Lysol. It's like a cleaning mechanism. It's intracellular digestion. So really what it does is that it cleans the cell by eating stuff that are dying or stuff that we don't need. So intracellular digestion, recycling cells materials, and then programmed cell death or apoptosis. So what it does is that it also programs when the cell is going to kill itself. And it can change these enzymes, and the reason why it has these hydrolytic enzymes is it can eat stuff that you don't need in the cell anymore. So here's a cell, here's another microscope image, and these little dots right here are called the lysosomes. So what it does, here's my lysosome, is that if I have something that we don't need or we need to get rid of, what my lysosome does is basically eat things that we don't need anymore. So here's a food vacuole, and let's just say the food vacuole is dying. It's a part of the cell that's not needed anymore, and lysosome just comes out and eats it. So it's essentially a cleaning of the cell. This one's called phagocytosis, where in this particular case you see a food vacuole coming in. The lysosome sort of merges with it and starts to digest all the stuff that's in the food vacuole. Autophagy is when I have a lysosome, and you might see it sort of phagocytosis is with a food vacuole, and autophagy, this one is for an actual body part. So I don't know how to explain this even more easily, but like phagocytosis, like I'm trying to eat food, which is a normal process. Autophagy, like the prefix auto, meaning it's trying to eat itself. Like there's things that it doesn't like, there's things that are bad, there's things that are dying in the cell. So the lysosome eats its own body parts because it's not useful anymore. So that's a lysosome. Vacuoles, they are basically the giant closets of the cell. So it's where it stores stuff. Plants have a super large central vacuole. Plants, because they don't move, they just store energy and they store food at a large rate. So we store food, water, minerals, pigments, poisons. Vacuoles are big storage areas. They're membrane bound. They have their own shell. There are different types, like a food vacuole, stuff like that. And again, if this is a plant cell, the vacuole for a plant, it almost takes up almost the entire cell. Like it's a big storage area. Okay. So just to repeat, if you're a visual learner, I have a picture here of a nucleus, the purple, and I have smooth and rough ER. And I want to make a protein. So the smooth ER and the rough ER, the ribosomes on top of it make a protein, and using a vesicle, like a delivery truck, it goes into the cis side of the Golgi apparatus. The Golgi's whole job is to package like a UPS, and it ships it out through the trans side. A Golgi can make a protein to be shipped out of the cell, or it can make its own lysosome, this purple thing right here. If you remember, a lysosome, its whole job is to eat stuff and clean the cell. And then some of these vesicles, they make these proteins so that other places can use it, like outside of the cell. So here's a vesicle from the Golgi that leaves the membrane. A full picture of what we're talking about. We don't have to go through the whole thing, but here are some full pictures of the cell. So hopefully you understood the endomembrane system. It's not supposed to be complicated, just that there's so many parts to it. Rough and smooth ER that have ribosomes, a Golgi apparatus, a lysosome, and again, which we didn't cover, but a super large vacuole. This is the one right here in plant cells that is used to store stuff. So hope you enjoyed, and I'll see y'all later.